For the first half of the 20th century, clinical nutrition focused on identifying and correcting deficiency. The primary targets were classical syndromes, like the severe gum inflammation of scurvy, where a single missing nutrient produced a predictable disease. By the 1960s, the focus shifted to population-level epidemiology. Food was reduced to macronutrient ratios and simple caloric math, primarily used as a tool for weight management and chronic disease association. This reductionist model assumes that identical calories and macronutrients yield identical biological effects across all patients. In complex metabolic cases, this assumption is the primary barrier to effective treatment. High-resolution clinical data now refutes the caloric model. When given the same nutritional inputs, different patients manifest categorically different metabolic and reproductive outcomes. The reductionist model fails to explain why identical meals produce divergent results. Restoring metabolic health requires a framework that treats nutrition as a high-fidelity informational signal interacting with a biological network. This systems biology approach identifies food as pharmacologically active information rather than a passive caloric substrate. Dietary components function as environmental signals. They are the mechanisms by which external inputs write instructions into the human genome and endocrine system. These nutritional signals target 4 critical nodes: the epigenome, the gut microbiome, peripheral circadian clocks, and the immune system. To understand the impact on health, we must trace the molecular architecture of this transduction from the nutrient sensors inside the cell to the epigenetic imprints passed across generations. Navigating these precise molecular pathways allows clinicians to address the network disruptions behind conditions like PCOS, rather than merely managing the outward symptoms. At the cellular level, the process begins with active surveillance. Cells do not passively absorb calories, they continuously survey their nutritional environment. This molecular seesaw represents the cell's internal nutrient sensing hub, anchored by 2 opposing enzymes, mTORC1 and AMPK. mTORC1 is activated by amino acid abundance and insulin. It initiates anabolism, driving cellular growth and protein synthesis. AMPK functions as the energy sensor, activated by cellular stress and nutrient scarcity. It shuts down growth to initiate catabolism and cellular recycling, a process known as autophagy. In a state of chronic nutritional excess, the mTORC1 pathway becomes hyperactivated, effectively jamming the seesaw in the growth position. This signaling failure directly impairs insulin receptor function, triggering the metabolic cascades observed in obesity and PCOS. Metabolic disease originates at this cellular level, when nutrient sensors lose their capacity to shift between abundance and scarcity signals. Within the nucleus, these signals translate into physical changes in gene expression through the genomic layer. Vitamins are not just metabolic cofactors, they provide the physical building blocks necessary for turning specific genes on or off. The 1-carbon metabolism network is the primary route for this process, relying on dietary folate, choline, and vitamin B12. These nutrients are required to synthesize S-adenosylmethionine, or SAM. This is the universal methyl donor. In DNA methylation, a SAM molecule transfers a physical methyl group to the DNA strand. This physical tag silences the gene without altering the underlying genetic sequence, acting as a molecular switch for the genome. A dietary shortage of these methyl donors prevents enzymes from marking developmental loci correctly, leaving genes unsilenced when they should be dormant. Without these specific dietary substrates, the epigenome physically lacks the materials required to execute its regulatory instructions. In the gut, the microbiome acts as a chemical processing plant that translates fiber into host signaling molecules. Commensal bacteria ferment dietary fiber into short-chain fatty acids like butyrate. These molecules cross the intestinal barrier to reach human cells throughout the body. Once inside, butyrate functions as a potent histone deacetylase, or HDAC, inhibitor. By blocking the HDAC enzyme, butyrate prevents DNA from spooling tightly. This forces the chromatin into a relaxed, open state. This open chromatin structure enables the expression of anti-inflammatory genes and programs the immune system for tolerance. The biological utility of a nutrient is not inherent to the food itself. It depends on the microbiome's capacity to bioactivate these chemical signals. The timing of nutrient intake dictates its metabolic impact. This is the temporal layer of chrononutrition. Feeding acts as the dominant time cue, or Zeitgeber, for peripheral clocks in the liver, gut, and pancreas. These peripheral clocks are driven by a rhythmic cycle of protein expression, anchored by a pair called clock and BMAL1. The act of eating rapidly shifts the cellular metabolic ratio of NAD+ to NADH. This metabolic shift is detected by SIRT1, an enzyme that monitors the cell's redox state. SIRT1 stabilizes the clock BMAL1 structure, physically resetting the organ's internal clock to the time of the meal. Because genomic accessibility shifts throughout the day, eating at 8 a.m. activates a completely different genomic landscape than eating at 9 p.m. Late-night feeding disrupts the alignment between the clock and the genome. Identical calories carry a higher metabolic cost when consumed out of sync with circadian rhythms. These molecular mechanisms scale across generations through the developmental origins of health and disease, or DOHAD. During early embryonic development, the genome undergoes a massive erasure and reestablishment of its epigenetic marks. This massive reprogramming is dependent on the immediate supply of methyl donors from the maternal diet. This photograph from the Dutch hunger winter anchors the event that proved nutritional stress in utero programs the metabolic set point of the offspring for life. Scarcity or excess during this window writes a lasting metabolic memory. Nutritional transduction does not stop with the individual. It creates an intergenerational signal that determines the disease risk of future generations. Because of these intersecting layers, standardized population-level dietary advice often fails in clinical settings. Continuous glucose monitoring at the Weizmann Institute of Science shows that metabolic responses to identical foods vary by individual, governed more by microbiome composition than by the nutrients themselves. This digital simulation shows how identical meals generate different outcomes. One profile produces a flat glucose response, while another generates a severe insulin spike. Effective management of endocrine and metabolic disorders involves moving beyond caloric math to account for this individual biological variability. Every meal is an active informational input that is continuously talking to your genome.